In this section, we're going to see how you get energy from food. So let's take a slice of pizza, for example. In the pizza, you have the crust, which the crust is a lot of starch, which is carbohydrate. You also have the protein. Pepperoni and cheese both have protein in them. And you have fat. The cheese and the pepperoni both also have lipid or fat. So when you eat a pizza, you're getting carbohydrate, protein, and lipid. Your digestive system is going to break these nutrients down. Remember chapter 2, polymers and monomers, you're going to break them down. When you break down and digest carbohydrates, you end up with glucose. Glucose is blood sugar, and here we have a picture of glucose. Glucose is going to be used for energy. When you digest the proteins, think back to chapter 2, proteins are made of amino acid. So your digestive system will turn the proteins into amino acids. You'll have amino acids in your blood. Now you can use amino acids for energy, but we're not going to get into that. When you digest lipids, one of the main things you get is triglycerides. And here is a picture of a triglyceride. And you can also use it for energy. So fats and carbohydrates are the main things you use for energy. And if we look at the triglyceride and the glucose, what they both have is hydrogen. Notice all these H's, all these hydrogens here in the glucose, hydrogens. So hydrogen is the main thing that you're going to use to get energy. So you eat food, you digest it into your monomers. They go into your blood. Your blood takes them to your cells, and then your cells are going to use the hydrogen that is in them to make ATP. Remember, ATP is the energy currency of cells. Then ATP will provide the energy for cellular work. So let's look at ATP now. ATP stands for adenosine triphosphate. So we have an adenosine. We're just going to put a big A here to represent the adenosine. Tri is 3, so we have 3 phosphates attached to it. So P represents phosphate. The lines represent the bonds, so these are bonded together. So these 2 lines here represent the bonds that are holding the phosphates to the adenosine. This one that's all curly like a spring is a high energy bond. So if you break this bond, a lot of energy is released. When your cell has work it needs to do, it breaks this bond. And when it does, you get ADP. So if you break that bond, the phosphate is no longer attached. It's just over here by itself. And that energy that was in the bond is released. So that energy is released, and it does work for the cell. Now you have ADP and PI, just a phosphate by itself. Well, now you recycle. If you think of this like your cell phone, this is your phone after it's been plugged in, and your battery is at 100%. You are set, you are good to go. Then you use your phone. So now here is your phone, and you're getting that little red warning that says battery low. So this is your battery is dead. So what do you do at the end of the day when your phone's battery is dead? You don't just toss your phone in the trash. You plug it in and you recharge it. That's what your body does with the ADP. You're not just going to throw it away, you just recharge it. So all you do is reattach that P. You create a new high energy bond and you reattach it. It's just like plugging your phone in and recharging the battery. Hydrogen is your charger. That is what you use to do this. So you use hydrogen that you get from your food. Remember, it's in the glucose, it's in the triglyceride. So think of hydrogen as like your charging cord for your phone. That's what you use to recharge this ADP and P back up to ATP. So now let's start looking at how this happens. And if you notice in your book, in your binder, it says like round 1, and then in a few pages you have round 2 and round 3. This is a topic, it's a very complex topic. It's covered in every biology class just about, and it's just one of those things that really gets students down. So I've broken it down into little bits, and I take a simplified view of it. So you may have learned a lot more detail and complexity in other classes. I try to slim it down to just what you really need to know to understand what's going on here. So cellular respiration is the process of how you use hydrogen from your food to take that ADP and PI and convert them to ATP so that your cell can have energy to do its work. So you have a cell. Here's a cell. And you may know from other biology classes, mitochondria is the powerhouse of the cell. That's where ATP is made. So we're going to give our cell a mitochondria. And you have blood. Remember, all of this nutrients are being carried in your blood. So here is your blood. And we're going to focus on glucose. Glucose is the main thing you use to make ATP. And the chemical formula of glucose is C6H12O6. So we have blood and we have glucose in it. You also have oxygen. We're going to put 6 oxygens here. So you're eating food, you're getting glucose, you're breathing, you're getting oxygen. These two things go to your cell. So your cell is going to take in the glucose. We're going to have glucose right here. It's going to go in the cytosol next to your mitochondria. Now remember, the overall goal is you want those hydrogens. First thing we're going to do is break the glucose in half. And when you break it in half, you're going to get pyruvates. Those are then going to go into the mitochondria. You're going to keep breaking it down. You're trying to get at the hydrogen. So you're going to break it all the way down so that you end up with just 6 carbons, 6 oxygens, and your 12 hydrogens. It's all broken up. Then you can use the hydrogens to make ATP. So you're going to have in your cell ADP and PI. They're going to come into the mitochondria also. You will use the hydrogen to push them together. So it's like you have to take your phone into the mitochondria because that's where the charger cord is and you charge it back up. You're going to use the hydrogen, push those together so that then ATP is produced. Then you're done with these, with the carbon, hydrogen, and oxygen. Your cell has to have a constant new supply. It can't reuse the glucose. So once you're done with those, oxygen is like the garbage man. It comes in, it's the cleanup crew. The oxygen binds to the carbon and the oxygen and the hydrogen from the glucose. And when it does, it produces carbon dioxide and water, 6 of each. Those are the waste products then that come back to your blood. So that's kind of the basics, the big picture of it. And with your ATP here, it would go to a place in your cell where you have work to do. You would break off that 3rd phosphate, release the energy to do work, and you would be back to having ADP and PI. So it's this ongoing cycle. So let's look at this in a little more detail now. We're going to break this up into steps. So we're going to have step 1 here is glycolysis. Glycolysis occurs in the cytosol next to the mitochondria. And this is where you have your glucose. So we have the C6H12O6. And we're going to break it in half. When we break it in half, we pull 2 hydrogens out. So first we pull out 2 of those hydrogens. And when you pull out hydrogens, they can't just be on their own. They have to be put on a carrier. So we have a carrier molecule called NAD. Just think of it like a taxi cab. So we've got a little taxi cab here. That's NAD. NAD is a taxi cab. It's a carrier molecule. It comes along to pick up those 2 hydrogens we just pulled out. So then we get to NADH. So that just means now there's a little hydrogen riding around in that taxi cab. Then we break what's left in half. So if you take C6, break it in half, you get C3. We had H12, but we took 2 hydrogens out. So that leaves us H10, and half of 10 is 5. O6 split in half is O3. We're going to have 2 of these. These are the pyruvates. C6H5O3 is pyruvate. When you break the glucose in half, it does release a little bit of energy, and you have a net gain of 2 ATP. Now, it actually releases 4 ATP, but breaking it uses energy. You have to use 2 in order to break it and get 4. So your net gain is 2 ATP. That's glycolysis. Take a glucose, you pull 2 hydrogens out, put them on your carriers, break it in half, you get 2 pyruvates, 2 ATP. So at the start of glycolysis, you had a glucose. That is what you started with. At the end of glycolysis, you have 2 NADH, 2 ATP, 2 pyruvates. That's what you have at the end. The next 2 steps I'm going to lump together here. We're going to have formation of acetyl-CoA and Krebs cycle. One of the things with biology is there's a lot of different terms for the same thing. What happened is hundreds of years ago, somebody named something. Then in more modern times, somebody decided to change the name. But then you have some textbooks use the old name, some textbooks use the new name. And so we end up just with multiple names for the same thing. Another name for Krebs cycle is citric acid cycle. In this stage, the pyruvates enter the mitochondria. And you're going to finish breaking them down and extracting the hydrogen. Remember, the hydrogen is what you want. That's your charger. So we have C3H5O3. We have two of those. These are inside the mitochondria now. We're going to pull out all of the hydrogen. So 5 here, that's 10 hydrogens get pulled out. Well, remember, whenever you pull out hydrogens, they have to be on a carrier. So we need 10 more little taxi cabs to come along and pick them up. We're going to have 8 NAD and 2 FADH. Just think of these as like different taxi companies. Think about if you go to an airport or a train station or something and there's different types of taxi cabs. That's what this is. NAD and FADH are just two different types of taxi cabs. They come along to pick up those 10 hydrogens. So you get 8 NADH and 2 FADH2. All of your hydrogens are extracted now. So we have all of the hydrogens on these carriers. Remember, we have 2 up here in glycolysis, 2 NADH, another 8 NADH, and 2 FADH2. So in the next step, we're going to use those hydrogens to make the ATP. But right now, we have to deal with waste. The hydrogen is the part you want. We have these carbons and oxygens. We have 6 carbon atoms, right? 3 here, 3 here. We have 6 oxygen atoms, 3 here, 3 here. They are waste. Think about when you go to the store and you buy a new pair of shoes. They come in a shoe box, and there's the box and there's tissue paper in there with them. Well, which part is it you actually want? Is it the box, the tissue paper, or the shoes? You spent all that money to get the shoes. The box and the tissue paper are just the packaging that your shoes come in. It's the same idea here. Hydrogen is what you want. Hydrogen is the shoes. It's what your cell is after. The carbon and the oxygen are just the box and the tissue paper that the shoes come in. So when you get home, you take your new shoes out and you put them on your feet, and the box and the tissue paper you throw in the trash. Okay, the carbon and the oxygen are waste. But you have to be careful of what you put in your blood. You can only put molecules in your blood that are safe to put in your blood. So this is where that oxygen comes in. If we can add 6 more oxygen atoms to this, then we could have 6 carbons, 12 oxygens. Well, that would make 6 CO2 molecules, and CO2 is safe to put in your blood. So you take 3 oxygen molecules to give you those 6 more oxygen atoms. So this is O2. That means it's a molecule atom bonded to an oxygen atom bonded to an oxygen atom. So for each O2, you have 2 oxygen atoms. So if you just take 3 times 2, that's 6. You get 6 oxygen atoms. This is basically saying you have 3 pairs of oxygen atoms. Well, if you have 3 pairs of oxygen atoms, you have 6 oxygen atoms. So we get 6 CO2. And this then is what you put in your blood. Okay, so you bought your shoes, you came home, you took them out of the box, you threw the box and the tissue paper away. That's what's going on here. Your cell got its glucose, It broke it up, it got the hydrogen, the part it wants, and it has thrown the carbon and oxygen, which we're just packaging, away. And your blood is where cells throw stuff away to. Now we're ready to go to the next step. Our next step is electron transport. This also takes place inside the mitochondria. And you have, it's called the electron transport chain. It's kind of like a slide. I think of it as like you go to the park and there's a sliding board. That is the electron transport chain. Back over here where I said those ADP and PI go back into the mitochondria, when they do, they go to the electron transport chain. So right here, we're going to have ADP and that P by itself. This is where the carriers take the hydrogen. So we have our 2 NADH here, plus 8 NADH here. That gives us 10 NADH. That comes up here to the very top of the electron transport chain, and it drops off the hydrogen. Remember, they're like a taxi cab. They pick up the hydrogen, they take it somewhere, they drop it off. So now the hydrogen is here at the top of the slide. The hydrogen goes down the slide. As it does, it pushes these together. So you get ATP. And then you end up with your hydrogen at the bottom of the slide, and you got 3 ATPs. Notice you have 10 of them. So 10 times you're going to do this. So by the time you do that 10 times, so you have 10 hydrogens, each one makes 3 ATP. You're going to get 30 ATPs made from those NADH dropping off their hydrogen. Okay, we also have our FADH2. It's the other brand of taxi cab. It drops its hydrogen off right here. Now notice it's not at the very top. It's kind of part way up. So this hydrogen, when it goes down, it makes 2 ATP. So we have 2 of these, and they each make 2 ATP. So that gives us 4 ATP. So the FADH misses that top one. It starts part way up the slide, and it makes 2 ATPs for each hydrogen it drops off. So this means altogether, when all 12 hydrogens go down that slide, you get 34 ATP. So electron transport produces 34 ATP for you. Now you have 12 hydrogens at the bottom of the slide. Remember we said hydrogen is like shoes. You buy a new pair of shoes, you wear them around for a while. Well, eventually they start to wear out. They get old and smelly, right? Like you're not wearing the same shoes now that you had 10 years ago. You buy new shoes now and then. Well, these hydrogens, by the time they get to the bottom of the slide, they're all old and smelly and worn out. It's time to get rid of them. So these are now waste. You've made your ATP, you're done with them. So again, you take in oxygen. Notice back here, I put 6 oxygens in the blood. We used 3 of them here. Well, this is where the other 3 come in. If you have 12 hydrogens, and remember 3 O2s, that's 3 pairs of oxygen, so that's 6 oxygen atoms, you can make 6 molecules of water. And this is called metabolic water. It's metabolic because it is made by your cells. So whenever you're doing cellular respiration, your cells are actually making water and putting in your blood. So this goes to your blood. So that is why over here we had 6 carbon dioxides and 6 waters going back to the blood from the mitochondria. So now let's summarize this. Let's put it all together, the whole big picture. You started off with C6H12O6. You also had 6 oxygens in the blood. We end up with 6 carbon dioxide and 6 water. Right? We're just looking at what was originally in the blood that leaves the blood and goes to the cell, and then what comes back to the blood. Now what goes in has to equal what goes out. So let's check our math and make sure we did this right. We're going to have carbon, hydrogen, and oxygen atoms. On the left, so this is what leaves the blood and enters the cell. And on the right is what leaves the cell and goes into the blood. We need to make sure these two sides equal each other. So carbon, here we have C6. So that is 6 carbons. Here we have H12. That is 12 hydrogens. Here we have 6 oxygens and another 6 pairs of oxygen. So this is 6. 6 pairs is 12. 6 and 12 is 18. So we have 18 oxygen atoms. Over here, 6 CO2. It's going to be 6 carbons. Here we have 6 waters. Well, each water has 2 hydrogens. So if you take 6 times 2, that's 12. 6 CO2, there's 2 oxygens in each of those, and we have 6 of them. So that's 12 oxygens there. 6 waters. Each water has 1 oxygen, and we have 6 waters, so that's 6. 6 and 12 is 18. It all adds up. So all you're doing is converting glucose and oxygen into carbon dioxide and water. The reason you do this conversion is your payment. What does your cell gain by doing this conversion? Well, it gets 36 ATP. We had 34 ATP here at electron transport. We had 2 ATP here from glycolysis. So that gives us 36 ATP altogether. You also get heat. If you look at page 73 in your book, it talks about the second law of thermodynamics. Whenever you convert energy from one form to another, you get heat. We were converting energy. Glucose is a form of energy. ATP is a form of energy. We converted glucose energy to ATP energy. Therefore, we also produce heat. So this is where energy comes from. This is where body heat comes from. So you can kind of think of this as your cell's job is to convert glucose and oxygen to carbon dioxide and water. And just like how you get paid money for doing your job, your cell is paid 36 ATP and heat for doing this job. So think about this. Let's put some implications to this. You get hot when you exercise and you shiver when you're cold. Well, whenever you use ATP, in order to make ATP, one of the payments you get is heat. So the more ATP you make, such as when you exercise, the more heat you produce. So when you exercise, you need more ATP. Therefore, you make more heat. And this is why you sweat. You sweat to get rid of that extra heat. Because remember, homeostasis, you want to stay at 98 degrees. Now, if you are cold, you will shiver. Shiver means your muscles are working. If they're shivering, they use more ATP. And if you use more ATP, you're going to make more heat. And this will warm your body up to homeostasis. Now let's look at some types of chemical reactions. We have some overlapping terms here, different terms that can be applied to the same reaction. So you already learned about hydrolysis and dehydration synthesis. Remember, that was with the you are what you could eat lesson. So you also know about polymers and monomers. In the top picture here, we are starting with a polymer. Notice it's two individual monomers linked together. We're going to break that bond and end up with separate monomers. So if we use a term you already know, this would be an example of a hydrolysis does this. Hydrolysis is a type of decomposition. So if you think of decomposition, it just means something breaks down. Decomposition is any time that you are breaking the bonds. So hydrolysis is a type of decomposition. It's decomposition in which you use water. Then we have the word catabolism. Metabolism is the chemical reactions of your body. And metabolism can be split into two types, catabolism and anabolism. So when we use the word catabolism, it again means breaking down. It's a type of decomposition. And in context of the body, like we talk a lot about protein catabolism, means you're breaking down proteins to get amino acids. Then we have exergonic. This has to do with energy. For the EX, think of exit. Exergonic, energy exits, or you release energy. We just talked about ATP and how when you break that high energy bond that holds the third phosphate on, it releases energy. That's an example of exergonic. So whenever you break bonds, you release energy. Then we also have exothermic. Think of therm. Therm means temperature. Remember the second law of thermodynamics we just talked about? When you use ATP, you get heat. Whenever you break bonds, you release heat. So you can tell that you have a decomposition reaction going on because temperature will increase. If one of these reactions is happening, the temperature will go up. It'll get hotter. Then the other side of this, we have synthesis reactions. So you already learned in the You Are What You Eat lesson about dehydration synthesis. Anabolism, remember, is the other type of metabolism. So this refers to it happening in the body. And here we have our monomers that we're starting with, and we are building a bond. So we end up with a polymer, and there is the new bond that we just made. And whenever you do this, when you're building bonds, that is endergonic. So think of the EN for enter. Energy enters. So you have to use energy to build bonds. And whenever you're using energy, that is endothermic. So now this time it's temperature enters. So you use up energy, you use heat. And because you're using it, heat is like money. Once you use it, you don't have it. So temperature goes down in these types of reactions. Another important thing to go along with reactions is enzymes. Enzymes are catalysts. This means they speed up reactions. So you have metabolism, chemical reactions in your body. You have catabolism, you have anabolism. These are constantly happening. They keep you alive. Enzymes speed them up. So you have all these reactions going on in your body. Remember, that is metabolism. Metabolism just means all the chemical reactions in your body. You need them to happen fast enough to keep you alive. Chemical reactions will eventually happen on their own, but it would be so slow that you would die waiting for them. So you need them to go fast. So you have enzymes in your body. The enzymes make the reactions happen fast enough to keep you alive. So enzymes are proteins that your body makes. You make your own enzymes to speed up your reactions. And you rely on your enzymes to stay alive. So we're going to look at enzymes and how they work. And it's important to keep in mind that they are proteins. Remember, proteins function based on shape. And remember, proteins can denature, which means change their shape and lose their function. If they get too hot, or if you change the pH. Right? Think about like cooking food. We talked before about a cooked egg versus a raw egg. Raw egg is clear and runny. You cook it, it's white and solid. You denatured the proteins in it. You changed their shape. So we look at an enzyme now. Here's our first example. Here's the lactase enzyme. It is used to break down lactose, which is the sugar in milk. So here we have a polymer. Right? It's the two molecules linked together. Notice the shape. They have a certain shape. The enzyme here has a shape that matches. This is the active site. So an enzyme has an active site. This is where the molecule fits. And enzymes are specific. Like how puzzle pieces fit together. Think about when you're doing a puzzle, you can't just put any two pieces together. Only the two that fit together can go together. It's the same with enzymes. Only lactose can bind to the lactase enzyme. Their shapes have to fit to each other. This is why there are people who are lactose intolerant. Some people don't have the lactase enzyme. And if you don't have that one enzyme, you can't digest lactose. You can't just use any old enzyme to digest lactose. Only lactase will digest lactose. So enzymes are specific to which molecule they will bind to. The molecule that it's going to bind to is called a substrate. So you have an enzyme and it fits to a substrate. So they bind to each other. So here you can see in this picture, the molecule is bound to the enzyme. This is called the enzyme substrate complex. Once they are bound to each other, the next thing that happens is the enzyme bends. Look at the shape of the enzyme. See it here? Compared to here, it bends. And this is not denaturing. It's bending on purpose. That's how it works. When it bends, it breaks that bond. So this bending is called induced fit. So it bends and breaks the bond. Now we have two individual monomers. Whatever is released from the enzyme is called product. And these are released. So this is now free to go into your blood from your digestive system. The enzyme goes back to its original shape, so it unbends. And then notice we have an arrow here that it goes back to the start. It's ready to go again. So you can reuse your enzyme. Let's look at another example. Here's our enzyme. And notice here, these are the active sites. Here is our substrate. Substrate is the molecule that fits in those active sites. And this is glucose. These are glucose molecules. Then here we have the enzyme substrate complex. So the glucose molecules are in the active sites there. They fit together like puzzle pieces. Here we have the induced fit. The enzyme bends. And this time when it bends, it forms a bond. And then we release the product. And then the enzyme unbends and it's ready to start again. So the basics here, you start with a substrate. Substrate is what you start with. It binds to the enzyme. The enzyme bends and it either breaks the substrate or links two substrates together, and you get product. Product is what you end with. And then the enzyme is ready to repeat. So you can reuse the enzymes, but there is a limit. They don't last forever. Like anything else, they wear out, and then your body needs to make new ones. So even though you do reuse enzymes, you also have to constantly make new ones. So they do wear out and you make new. So every day your body is making new enzymes. Another thing to think about with enzymes is they work by bending. That bending is key. It's when they bend that they form or break a bond. Temperature affects ability to bend. If something is cold, it is stiff, and it will resist bending. It's hard to bend something that is cold and frozen. If you warm it up, that makes it more flexible, and it will be easier to bend. This information is going to help you with your lab. Remember this, the effects of temperature on the ability of an enzyme to bend. And if we tie this in to what we had up here with types of reactions, this top one here, you're breaking bonds, so this is a decomposition. And the bottom one here, we're building bonds, so this is an example of synthesis. So enzymes work in both decomposition and synthesis reactions. Let's go back to cellular respiration now and add in some more information from what you learned before. So you'll recall that you have a cell, and your cell has a mitochondria, and you have your blood, and we take glucose and oxygen from your blood. They go into your cell. You do the whole glycolysis, acetyl-CoA, Krebs cycle, electron transport. You get your ATP. You get heat. And you end up with 6 carbon dioxide and 6 water going back into the blood. So what we're going to focus on in this lesson is that carbon dioxide that goes back into the blood. So every time you do cellular respiration, you put 6 carbon dioxides into the blood. We're going to follow this carbon dioxide and see what happens with it. Blood, as you may know, is mostly water. So we have carbon dioxide being mixed in with H2O. They react with each other. There's a chemical reaction you get when you put carbon dioxide in water. You already know this because of your Chapter 1 lab. You used your straw to blow into the bromethyl blue and you turned it from blue to yellow because the carbon dioxide you were blowing out was having a chemical reaction with the water in the beaker. These two molecules will combine with each other. H2CO3. All that is carbon dioxide and water together. So H2 comes from right here, C from the C, and you have 2 oxygens and carbon dioxide, 1 oxygen and water, that gives you O3. H2CO3 is carbonic acid. Now it doesn't stay like this. It dissociates. Remember, you've already learned about the word dissociate. We talked about like when salt mixes with water, it dissociates into sodium and chloride ions. Carbonic acid in your blood dissociates. One of the hydrogens breaks off. So one hydrogen leaves. Now remember before when we talked about hydrogens breaking off from water, hydrogen has 1 electron. When it breaks off from a molecule, it leaves its electron behind. So the HCO3 keeps that electron. So it has a negative charge because it kept the electron, and electrons are negative things. The H by itself has a positive charge. It no longer has an electron. It left its electron with the molecule. So it's out here without an electron. Electrons are negative. It has none of them, so it is positive. This one, HCO3- is bicarbonate. It's often called bicarb for short. And this is hydrogen ion. Think back to what you learned about pH. An acid is anything that increases the concentration of hydrogen ions. So because we're adding a hydrogen ion, this lowers the pH. This hydrogen ion results in reduced pH. So the effect is every time your cells do cellular respiration, they lower the pH of your blood. So your cells make ATP. When they make ATP, they put carbon dioxide in your blood. This reacts with the water in the blood, and you end up increasing the number of hydrogens in the blood. And when you increase the number of hydrogens, you reduce the pH. So this is tying in with what you did in the Chapter 1 lab. You first experienced this in Chapter 1 lab. Now we're just kind of formalizing it and giving you notes to go with that. Your blood has to stay at a pH of 7.4. Think about why it's dangerous to change that. You should know by now. If you change pH, you denature proteins. Enzymes are proteins. And remember, you rely on your enzymes to stay alive. It's so important to keep your blood at 7.4 so you don't denature your enzymes. On one side, you have cells. Your cells are making ATP, and in the process of making ATP, they put carbon dioxide into your blood. Every time you put carbon dioxide into your blood, you get more hydrogens and your pH drops. Well, if you just let this go, your pH would drop, you would denature your enzymes, and you would die. So that's not a good option. Luckily, we have your lungs. Your lungs take the carbon dioxide out of your blood and exhale it. You have to maintain a balance. You want to keep homeostasis. Think about what would happen to pH if you took too much carbon dioxide out of the blood. If you were taking too much out, your pH would go up. So if you have too much CO2 in your blood, you have a low pH. That can kill you. However, if you have not enough carbon dioxide in your blood, you would have a high pH, which also could kill you. So you have to maintain just the right amount of carbon dioxide. So the amount that goes in has to equal the amount that goes out. So let's say, we should make up some numbers to make it easy. Let's say you have 100 carbon dioxides in. That means you need 100 out. Let's say then your cells start using a lot more energy and you have 500 carbon dioxides in. Well, that means you need to get 500 out. And in order to increase, to get 500 out, 400 more than you were before, is where you have to breathe deeper. And Your blood is what's transporting this from your cells to your lungs, and your heart is what pumps your blood, so you need your heart to beat faster. Which is exactly what you saw in your Chapter 1 lab. So, cellular respiration plays a big role in your pH. And your breathing rate, the reason you breathe heavy when you exercise is to get rid of that extra carbon dioxide so you can keep your pH at 7.4. A lot of people want to say, oh, you breathe heavy so you can get more oxygen. You do use more oxygen when you exercise, but that's not why you breathe heavy. You breathe heavy to get rid of the extra carbon dioxide and keep your pH at 7.4. Let's look at another aspect of cellular respiration now. When you are exercising full force, as much as you can, so when you are full out exercising, say you're running a marathon, right? You're giving it your all. You do use a lot of oxygen. Remember, for each time you do cellular respiration, you use 6 oxygens. So you do use a lot more oxygen. Your heart and your lungs do send more oxygen when you're exercising. Yes, you are breathing deeper and faster. Your heart is pumping faster, so you are sending more oxygen. But if you look at your muscles on one side, when you're exercising, your muscles use oxygen. So they are taking it out of your blood. And on the other side, you have your lungs and your heart are putting oxygen in. When you exercise as hard as you can, it's not equal. You don't have an in equals out situation. Instead, what happens is your muscles use more. There is more going out than what is going in. So the result of that is the amount of oxygen in your blood drops. So in this scenario, exercising as fast as you can, your muscles use more oxygen than your heart and lungs provide. The result of this is a decrease in your blood oxygen level. And you go into a state of oxygen debt. Once you're in oxygen debt, you can no longer supply oxygen to every cell. You have to start rationing. So let's look at what happens to a cell if it doesn't get oxygen. So remember, we have the cell, we have the mitochondria, you have your blood, and we'll put the glucose here. But we're not going to put any oxygen there to go into the cell, just the glucose. So if the cell has just the glucose and not the oxygen, it can still do glycolysis. Remember, glycolysis occurs in the cytosol next to the mitochondria. So we're going to take that glucose and we're going to pull out 2 hydrogens. We're going to have our 2 carriers come along to pick them up. Then we're going to take what remains, break it in half, so we get our 2 pyruvates. And we have a net gain of 2 ATP when we do this. Glycolysis can still happen just fine. Normally, those pyruvates would go into the mitochondria. But when there's no oxygen, they can't do that. Mitochondria shuts down. And the pyruvates can't get in. This reminds me of the National Lampoon's movie. So you might be wondering what these pictures have to do with anything. If you remember the National Lampoon's movie, they drive across the country to get to Wally World. So here are the Griswolds. They're the pyruvates. They're ready to get into Wally World. Just like the pyruvates are ready to go into the mitochondria. And if we skip up a little bit,
Sorry, folks, we're closed for 2 weeks to clean and repair America's favorite family fun park. Sorry. What?
Clark, what are you doing?
We watch this program.
so you get the point. The Griswolds are the pyruvates. This is the Griswold family. The mitochondria is Wally World. The Griswolds want to go into Wally World, but they can't because it's closed. The pyruvate wants to go in the mitochondria, but without oxygen, it's shut down. So, if you've seen the movie, the Griswolds are trying to get into Wally World, and a security guard, John Candy, comes out and says, "Sorry, folks, park's closed. Moose out front should have told you." So I always imagine a little moose standing here. So draw in a little moose here in front of the pyruvate. And the moose, or in front of the mitochondria, and the moose says, "Sorry, pyruvates, mitochondria is closed." So now what do? Your pyruvates can't go into the mitochondria. No electron transport. You're not going to get those other 34 ATP. You're just stuck here. And you have your NADH. Remember, this is a taxi cab. Imagine you're in a taxi cab and like you fly somewhere, you get to the airport, you have the taxi cab take you to a concert you're going to. Well, you get there and it turns out the concert is canceled. What are you going to do? You can't just stay in the taxi cab. You have to get out of that taxi cab. You have to find someplace else to go. Same thing here. The NADH is not going to keep the H. It's got to get rid of it. So you're going to put the H back onto the pyruvates. So now you get C3H6O3. This is lactic acid. So if you lack oxygen, you switch from cellular respiration to fermentation. In fermentation, you can only do glycolysis. You're not going to use your mitochondria. You're only going to get 2 ATP. Now remember before you got 36 because there were another 34 that came from electron transport. You're not doing that now. So your payment has gone way down. Remember I said this is like a sales job. Imagine you have a job that pays $36 an hour. Then one day you go into work and your boss is like, yeah, today I'm only going to pay you $2 an hour. That's not so good. Also, instead of the pyruvates going in and finishing being broken down, they get the hydrogen added to them, and you get lactic acid. Now, lactic acid is going to reduce the pH in your muscles. Enzymes don't just instantly denature, they slow down first. So this slow drop in pH is going to inhibit your enzymes. They're not going to work as well. So you're only getting 2 ATP. The pH in your muscles is dropping, which is making it hard for your enzymes to work, and you are going to stop exercising. So imagine I said, like, hey, I'm going to give extra credit to anybody who can hop up right now and run all the way to Peoria. And you have to sprint as fast as you can the whole way there. You can't stop. You can't take a break. You might give it a try, but chances are I'm going to have to come along and find you in the ditch on the side of the road. Unless you're an amazing super athlete, you're not going to make it, because your muscles are going to use oxygen faster than your heart and lungs can send it. Your cells are going to switch to fermentation. They're only going to get 2 ATP. There's going to be lactic acid building up, and you're going to stop. One interesting thing to think about is athletes, when you train, if you wanted to run to Peoria, it is humanly possible. You would have to train a lot. Think about what gets better when you train. Even if you didn't want to run all the way to Peoria, say you just want to run a 5K. If you train, you'll eventually be able to do it. It's your heart and lungs that get better. Athletes can keep running for a longer time than somebody's out of shape. Yes, their muscles are better, but it's not just the muscles. Their heart and lungs are better at sending oxygen, so they don't switch over to fermentation as quickly. Their heart and lungs are able to keep up with the muscles' demand of oxygen. So their cells don't switch to fermentation. And you can tell this if you know anything about heart rate, if you know about athletes' heart rates. An average adult resting heart rate is 75 beats per minute. An athlete's resting heart rate can be in the 40s, can be 40 to 50 beats per minute. That's an indication that their heart is really strong and really healthy and working good, because their heart is so efficient that it doesn't have to beat as much when they're at rest. So an average adult at rest, just to keep up with keeping the body alive, has to beat 75 times a minute. An athlete's heart is so good at sending oxygenated blood that when the athlete is rest, their heart only has to beat 40 times a minute to keep the body going.